Hello, hello, hello. There we go. Good morning. This is Void Biology, and this is Cellular Respiration Part 1. Here we go into a different, really important organelle, the two organelles we've been focusing on for quite some time. These independent, autonomous, mostly autonomous city-states, right? They have their own DNA. They're so complicated and essential that they have their own set of instructions. Don't need to take direct instructions from headquarters. They, of course, benefit from all the other activities occurring in the cell, as this is a very interdependent type of system. It's really like an ecosystem here in the cell, all these important parts that are involved with one another. Peripherally, some directly, and we've been spending a lot of time on the chloroplast. It's time to get down into the detailed mechanisms happen in the mitochondria. Our discussion has been about metabolism. And so we'll start talking a little bit about metabolism, reminders about what this is here, and also thinking about our criteria for a creature being considered alive. A creature that is living can do a set of things that we've listed off before. The same can be said for a cell. A cell can do all of these things. And we've talked about some of those characteristics already. We've talked about the ability to sense and respond to their environment. Cells sense and respond to their environment. We've talked about their ability to metabolize, and that's what we're continuing to talk about today. And then coming up soon, we'll be talking about their ability to reproduce. So cells are capable and do reproduce, not just single-celled creatures, but all of your cells, most of them are able to and do reproduce themselves. And so here we go. Let's head to the world of cellular respiration. I really like this image here. This is of course, the mitochondria. There it is in all its glory, and some fabulous fluorescent staining technique here. Look at this. Isn't this incredible? And so you can see this mitochondria, its double membrane jumps out at you. You can see its inner matrix. You can see this inner membrane that's folded, forming what we call the cristae. And so anatomy that has some similarities to the chloroplast, a double membrane. But this inner membrane in this case is folded to form the surface area where much of this reaction takes place, as opposed to the thylakoids being present inside the chloroplasts. So here's the mitochondria, and the cells have various numbers of mitochondria. I was listening to a student's presentation earlier today about the remarkable cuttlefish, and the cuttlefish has these specialized cells called chromatophores in their skin, embedded in their skin. And these chromatophores have the incredible ability, along with some other cells that do some reflecting type of and color combining. These chromatophores help cuttlefish and squids and octopus to camouflage. They require a ton of energy to be able to do that. So hundreds of mitochondria or more in these cells. Muscle cells have a lot of mitochondria. Many other cells do not need so many mitochondria. That's not the role of that cell. Isn't necessarily to produce surplus energy. In other words, all cells need to produce energy for their own processes. But if it's a cell that's not required for sending energy out, then that cell is going to have fewer mitochondria. And so cellular respiration, it is this process of making all the chemical energy needed for that cell, and then some, depending on its specialty. And this ATP, we'll review its structure again here in a moment. This illustration, this micrograph, this is an electron micrograph from a news site that's reporting on an important cellular disease called Fragile X syndrome. Fragile X syndrome implicated in autism and different types of intellectual developmental problems in people. And this, it's thought that one of the more recent studies is showing that the mitochondria might be the source for this problem. So some problems with the membrane structure in the mitochondria that doesn't allow for enough ATP production. So an interesting connection there. There are a lot of cellular diseases, and we're learning a lot about cellular diseases. And you can kind of think about the insidious nature of a disease being cellular. It's really hard to heal a disease. It's going to take a different toolkit to attack a disease that's cellular because they are reproducing. It's not like you have a virus or a pathogen in your body that can be gotten rid of. This isn't a problem that is integral and kind of built into the cells. So the problem stemming back somewhere during initial development, probably, which we're going to be heading that direction. So metabolism, what is metabolism? It's really the collective whole of all the chemical reactions that are related to energy production and use. The collective whole of the cell's chemical reactions, really especially related to energy production or use. And so each cell has its own metabolism, of course, and that's the export and import trade that we've talked about before a little more broadly. Okay, so there's that energy currency of the cell, ATP, adenosine triphosphate, adenosine being what we call that nitrogenous base plus that ribose sugar. You should notice nitrogenous base making up nucleotides, a nucleotide being the monomer for nucleic acid, nitrogenous base plus a sugar plus a phosphate. That's your typical nucleotide. Then we saw stacks of those, rungs of those, stacked, making up the DNA molecule. And of course, we'd see a hydrogen bond here linked to another nucleotide. And so adenine is the nitrogenous base that we see in ATP. It is a nucleotide, has a different function though. And its sugar is ribose. So it has the same sugar as what nucleic acid? Ribonucleic acid, RNA. And it has 3 phosphates. And that third phosphate, this is the energy releasing bond right here. So this phosphate gets subtracted and reattached continually. I read an article recently that suggested that each ATP molecule turned over was recycled. The P was broken off, ATP broken off into ADP and P over 1,000 times per day. All right, so the turnover of ATP, I often get questions like, how much ATP does the human body produce in pounds or kilograms? That's hard to quantify, mostly hard to quantify because the turnover is so fast. It's kind of like stocking the shelves and then, a few seconds later, they're empty. Then stocking the shelves. So stocking the shelves 1,000 times a day, right? That's hard to quantify your inventory. And so that is how much ATP gets used, and that's how integral it is to every process. So if there's an ATP production problem, like Fragile X syndrome, that's going to be significant problem there. So a closer look at what happens to ATP, we call this phosphorylation. Phosphorylation is adding that P back after it's been broken off and the energy is released. And so if we look at this slide here, here's our ATP molecule, adenosine triphosphate. This phosphate breaks off, releases energy, and that's how the energy is converted or passed on at the correct location so other chemical reactions take place. Any active transport that needs to be done requires ATP. Okay, and so when that phosphate pops off, energy is released, bond energy, that's the high energy bond. And to rebuild this ADP plus P, the P doesn't get dissolved, the ADP doesn't get dissolved, the energy is converted. And then to bring this P back into ADP to form ATP again takes a lot of energy. And so that's the phosphorylation. The phosphorylation of ADP is simply adding a third phosphorus. And they also call that oxidative phosphorylation. You'll see that here in a little bit. So phosphorylation, just think adding a phosphate, okay, which is PO4 negative. Okay. And that phosphate will often bond with another molecule temporarily. So that does often occur, but that tends to not be the final step. That energy might be passed on, and that phosphate might actually bond with another chemical, but it's not going to stay there. Okay, so formation of ATP, that's what cellular respiration is all about. Here's another quick look at the ATP cycle. And we see here, you can get ATP energy from almost anything. The liver, our digestive system, the human body is incredibly capable at taking any type of energy source that we eat, proteins, lipids, or carbs, and turning the right amount into ATP, storing the right amount. If we eat a balanced diet where we get all of those in the close to the right quantities or ratios, we're going to be the most healthy. But the body's really good at converting any of those into this chemical form, and that's going to be primarily by converting a significant portion of these to glucose. So we eat a whole bunch of food, carbs, lipids, proteins. They get digested, absorbed into the bloodstream in the small intestine. Some are going to go to the liver and stay there and get reformatted. Others are going to make it to the cells, and that glucose molecule will make it to the cells, and that glucose, along with oxygen that we're breathing in, that oxygen goes into our capillaries in the lungs at the site called the alveoli. Those are the two ingredients that need to make it down there to each cell for ATP synthase, for ATP synthesis to occur. So you're going to see some similarities between the last stages of photosynthesis and cellular respiration. This electron transport chain, this ATP synthase molecule that's fueled by hydrogen ions, that's found here also. So kind of an interesting exercise thinking about the complexity of molecular machinery here. Amazing stuff. Two types of chemical reactions. We call them exergonic and endergonic. Exergonic means energy released. And that is we're going to see that when ATP is reformed. ATP is reformed, ADP plus P, and some energy is released there. And then the hydrolysis or breakdown of ATP to release energy, that's considered an endergonic reaction. I'm not too worried about those terms, but just to give you some information there. Exergonic, releasing heat. You're going to feel that as heat, like a combustion reaction is exergonic. An endergonic reaction would be something like you'd see with one of those cold packs that you put in your first aid kit, those disposable instant cold packs. You break two chemical bag linings together and it starts to get cold. In other words, it's absorbing all the heat from its environment. It's getting colder. It feels cold. That's an endergonic reaction. Or exothermic and endothermic. Those are appropriate terms too. Okay. So our goal here in our mitochondria is to extract energy and then convert it. All right, so the glucose comes to the cells, the oxygen comes to the cells. We've changed that those carbs and lipids and complex molecules, complex polymers, and they've been turned into glucose. And now it's time to convert them into a different form of chemical energy. So cellular respiration is all about chemical conversions. It's not the glamorous step of converting electromagnetic radiation into chemical energy, which is really a miracle. This is converted. This is still amazing stuff, but we're staying in the chemical realm, right? We start off with chemical ingredients and we're converting this matter and energy to different types of chemical ingredients. So a little review there. Combustion reaction is one that burns fuel in the presence of oxygen and produces CO2 and H2O primarily. There are going to be some other products in many combustion reactions. None are perfectly efficient. None do perfect or complete combustion. In a wood stove or a car, you'll often get a CO. In a car, you'll also get some sulfurous and nitrogenous types of gases produced. We were recently visiting a steam plant here where we live, and they had a remarkable process where they were able to release almost zero emissions and convert almost all of the product, in addition of producing water, almost all of it into ash. And so really amazing. They were able to manipulate the amount of oxygen coming in and the heat regulation to such an extent that almost all their product was ash and H2O, which is just fabulous. Really thoughtful stewards, not only thinking about unnecessary gaseous emissions, but also thinking about, hey, how can we make something, a waste product, reusable? And so the different agricultural departments in colleges use that ash as a soil amendment. So pretty cool. Pretty cool process there. So combustion, we know that one well. That's how we generally talk about cellular respiration. burning glucose in the presence of oxygen and producing CO2 and H2O. Redox reaction is one that involves the reduction of a compound, reduction meaning adding electrons to something, and then oxidation means losing electrons. And so photosynthesis is a redox reaction. There are also some redox reactions that occur in cellular respiration. So we can't say completely that cellular respiration or photosynthesis is only redox. It also has some combustion reactions there. And there are many sub-reactions in cellular respirations as well. But overall, the big picture, the reactants and products make it a redox reaction. So we're going to be reducing an important, or we did reduce a really important reactant in photosynthesis. We reduced the CO2. That's the final electron acceptor. In oxidation, we're stripping electrons away. Those electrons are initially stripped from water. They're also stripped from chlorophyll. And then CO2, again, is the final electron acceptor. So CO2 is reduced because it gains negative electrons. Oxidation is the losing of electrons, and that's initially our water molecule. Third type is hydrolysis, which we've talked a lot about, and water being the universal solvent, hydrolysis being the opposite process from dehydration synthesis. So remember that process of adding monomers like glucose, add two together. When you add them together, water is released and the two molecules join. That's called dehydration synthesis. And that is the process of building up, or catabolism. Hydrolysis, the process of breaking down, or, and I think I misspoke there. Anabolism would be the process of building up. Catabolism would be the process of breaking down. So hydrolysis, water comes in and dissolves those bonds between the different glucose molecules, breaking them down into their monomers again. Okay, so two types of cellular respiration processes. Most living things really need oxygen and a lot of it. Plants need a lot of oxygen. Just because they make oxygen doesn't mean they don't need it. They definitely need it. And so when we talk about the two different types of cellular respiration processes, or energy production processes in the cell, we talk about mitochondria cellular respiration, aerobic respiration, means requiring oxygen. That's us. That's almost all living things. But there are some that actually oxygen is poisonous to them, some that can still do this process in the presence or in not in the presence of oxygen. And those are called anaerobic processes. And fermentation is our key example there. So fermentation does not require oxygen for cellular respiration to proceed, for ATP to be made. Now, important, interesting distinction here, or key fact about cellular respiration, is the first step can still happen. So if you're low on O2, think about this practically. You're going for a run. It's first day of cross-country practice, and you decide you're just going to run that 5K and kill it. And you run out of oxygen eventually, right? You're going to get cramps. You're going to slow down. You're going to have to walk. You're using up all your available oxygen, and that has consequences because cellular respiration can't necessarily continue. Glycolysis, the first step can, though. So you can do glycolysis without oxygen. So you can produce a tiny bit of ATP in the first step of cellular respiration without oxygen, but the rest do need it. And the rest are really the last step is the payoff step. We'll talk about economics a lot during this explanation of cellular respiration, because this really is a chemical accounting exercise. And it was so in photosynthesis also. We're counting certain things. Think about the things we could have been accounting for in photosynthesis. And why might we do that? Why might it be helpful or important or useful to do accounting? Well, because we know that this process exists in the known universe, and in our known universe, there are some rules that apply to everything. And so in our known universe, we have the conservation of mass and the conservation of energy. Energy and mass can't be created. or destroyed. And so our chemical accounting needs to add up on both sides of our equation. And so in photosynthesis, we could have accounted for what might have been interesting to account for, in addition, of course, to the C, the H, and the O. It could have been helpful or interesting to account for each electron or each H plus ion, the proton, those especially. And ATP is always an interesting one to try to account for. And so that's how we approach cellular respiration. Here we go. So this is that grand process, converting glucose to ATP energy, and it is the inverse reaction of photosynthesis. So instead of CO2 plus H2O, we've got glucose plus oxygen. So we're burning that glucose molecule in presence of oxygen, right? It takes 6 molecules of oxygen to burn that glucose. That's a pretty big molecule compared to these. And there is a net production of ATP. Now that's not really part of our balanced chemical equation, but that is an important net at the end. And that number is going to vary depending on what you read. And scientists are currently discussing that and not necessarily agreeing. Could be up to 38 ATP are produced, could be closer to 30 ATP are produced. There are some efficiency differences because living things, not that we're nearly as simple as machines, but like machines, we have certain things that can improve or decrease our efficiency. And so you're going to see that number vary a little bit. But we mostly want to just track and account for our ATP and our know where our reactants go and know where our electrons and hydrogen ions go. So glucose plus O2 yields CO2, water, and then our chemical energy. So there's 4 main stages. Actually, that's a little bit of a typo there. 4 main stages in, depending on your textbook, some will say 3. I like to call the middle step between 1 and 3, step 2. Some textbooks really don't want to add a 4th step for some reason. I think it's easier to think of it as a full step. And you'll see why hopefully in a minute. The first step occurs in the cytoplasm. First step occurs in the cytoplasm, and that's really important. First step occurs in the cytoplasm, and that's really important because the glucose molecule can't make it into the mitochondria, right? The glucose has to be dealt with outside of the mitochondria. The other 3 steps, 2, 3, and 4, take place in the mitochondria itself. So glycolysis, the breaking of glucose, happens outside of the mitochondria, the rest inside. So know the geography that goes with each of these 2. And here we go. Here is our cellular respiration slide that you'll get to know very well. I'll put these up as a raw material so you can access the slides. 4 steps. There's our mitochondria, the outline of it. Double membrane, folded inner membrane, the matrix. Here's the liquid inside, and the cristae are these inner folds. And so here comes glucose, and glucose gets split out here in the cytoplasm. Glycolysis, splitting sugar. Lysis, rupture of a cell. Glycose is being ruptured or split. That one glucose is going to split into what we call pyruvate or pyruvic acid. They're used interchangeably. Same formula. You don't need to know the formula, but you need to know that 2 3-carbon molecules of pyruvate are the result. So glycolysis gets split out in the glycolysis is the splitting of the glucose out in the cytoplasm into 2 pyruvates or 2 pyruvic acids. That gives us a net of 2 ATP. We'll account for these in a moment. And we see some release there of CO2. Oh, sorry, I'm getting ahead of myself. That's coming in the next step. All right, so cytoplasm, glycolysis, the splitting of glucose, and 2 ATP produced. Those pyruvates now are small enough to make it into the mitochondrion itself. So, and that's where these pyruvate molecules, these 3-carbon, these are organic compounds, are going to be oxidized. So remember what our definition of oxidation is. Oxidation is the stripping of electrons away. So pyruvate's electrons get stripped away. It's oxidized. Those electrons are really important. You know how important electrons are in powering these cellular processes. And so pyruvate gets oxidized. That gets rid of one of those 3 carbons. And so a little CO2 is released here. The CO2 diffuses out of the cells into the bloodstream, out into the capillaries, out into the alveoli, and we breathe it out. So any CO2 here is a waste product that's actually toxic. It's not like O2, the waste product we get in photosynthesis. The CO2 has got to go. It cannot build up or we have some serious problems. So CO2 leaves, and that's the oxidation of a pyruvate step 2. And no net ATP are produced here. So this reaction is a lot like a startup business. If you started up some business and you're making some widget for sale, and you've got some investors, you got some funding of your own to start up with, and you're breaking even early on at best. And so breaking even, you got net 2 ATP made here in step 1, you got 0 here in step 2. So year one and year 2 of your startup business, of your ATP production company, or whatever widget you're making, you're just scraping by. Now you're telling your investors, hey, we just need about 4 years and we're going to be super profitable. Not just profitable, but super profitable. We will not be in the red years 1 through 3, but we will very much be profitable by year 4. Okay, so step 2. Step 3 is called the Krebs cycle. Krebs cycle churns out tons of electrons, which we know are critically important for powering different things. The Krebs cycle used to be called the citric acid cycle. 2 cycles through, right? And so pyruvate gets oxidized. It turns into this other molecule we'll talk about in a moment. And we get 2 ATP here. 2 ATP are produced in the 3rd step. So net so far, 4 ATP after 3 years, right? Per year, that's how much of our widget we're producing. We're profitable, but not by much. Those electrons get carried by our friends, the electron carriers, over to the well-known and well-loved electron transport chain. You saw this in photosynthesis, and we know what that thing can do. And that thing churns out ATP. And in this case, it churns out a whole bunch of ATP. You're about to see how much in a moment. So 4 steps, 1 in the cytoplasm, the first, the second, third, fourth in the mitochondria itself, waste product of CO2, and a whole bunch of ATP being produced here. And I probably should have put this one first before that illustration. But here again, our double phospholipid bilayer, those 2 membranes, the matrix being the interior inside the inner membrane, and then the cristae just being those folds on the inside. Lots of surface area for lots of types of reactions to produce products that are easy to get out, but also there's a lot of miles of coastline to have factories. Think about surface area kind of like coastline. You got a lot of coastline, you got more opportunity for a port. A lot of coastline, you have more opportunity for cellular machinery to generate energy, or if you're in a chloroplast, of course, to generate some glucose. Okay. 2 more slides for today. That'll be our introduction, guys. So here you're seeing a very similar type of diagram to before, but a little bit more information. So all 3 of the steps look the same, but here we're showing that what we have on average for our net production of ATP. So glycolysis, the splitting of glucose in the cytoplasm, net of 2. Oxidation of pyruvate, 0 to 2, might get 0. Here in this step, that pyruvate gets oxidized into acetyl-CoA. So important to know what reactant goes into a step and what product comes out. Step 1, glucose goes in, cytoplasm. Reactant that comes out is pyruvic acid or pyruvate, same thing, 2 ATP. So you should be able to do that with each step. Name the step, reactant and product of each step, and how much ATP comes out. It's hard to get that fourth finger out. That pinky wants to come up too. So glycolysis, glucose to pyruvate in the cytoplasm, 2 ATP net. Oxidation of that pyruvate turns into acetyl-CoA. Electrons are stripped away, oxidation. And we have a net of often 0 ATP. So if you had to pick between 0 and 2, just pick 0. Third stage, that acetyl-CoA gets converted into a ton of electrons that get carried away down the electron transport chain, a net of 2 ATP. This is 2 turns. So kind of like Calvin-Benson cycle, you need 2 turns here per glucose molecule. And then fourth and final is that electron transport chain. All those electrons show up, all those hydrogen ions show up. And look at that. Our 4th year, we're profitable, Mr. Banker. Check that out. That we are killing it with 34 ATP. That's a lot. That's the payoff step. So these are the investment stages, and this is the payoff step. And this operates nearly identical to what we see in the electron transport chain in the light reactions in photosynthesis, except there aren't photosystems. Photosystems aren't, light's not providing the energy here, glucose is. But very similar type of movement of the electrons. We're throttling the energy down. We can't burn glucose, it'll kill the cell. And so we've got to throttle that glucose down into an electron stream that powers these various processes. And at the end, of course, our good friend, the ATP synthase molecule, which spins and spins and spins as hydrogen ions pump through it. That causes the spinning and the making of new ATP, the phosphorylation of that ATP, adding that P back to ADP. And so at the end of the day, see this in written form here. And this slide and the last one I say are probably the most important of this whole section, because they show us and then tell us what's happening each stage of cellular respiration. Again, you don't need to know the numbers of the products necessarily or the numbers of the electrons, but you do need to know the entering reactant, the leaving product, where it occurs, and then the net of ATP. So glycolysis, pyruvate, Krebs, and the electron transport chain give us really between 30 and 38. Some newer sources are saying that 36 to 38 estimate that textbooks have been saying for several decades is a bit high. I don't know how that's going to sort out, but I think it's always interesting and helpful for us to be humbled by newer information and just to hold things. We've got to hold our theories like this, right? Cell theory, very much a theory, very much being refined. Atomic theory, all of our big areas of knowledge in science, we can confidently say that they are in the theory stage. Theory is not like a theory or a hypothesis on the street. There's a lot of credibility there. Theory is a grand explanation for a whole bunch of disparate but related phenomena. There's a lot of scientific evidence supporting that theory. That doesn't mean it can't be wrong, but it does mean that it's doing a pretty good job of explaining what we know right now. Okay, so I hope you enjoyed part 1 here in cellular respiration. We'll get into a little bit more detail of that in fermentation in part 2, and we'll see you next time.